So don't apply yet, the jury's still out. Well, thank you very much. It's really exciting for me. I was just walking through the engineering buildings before I came over here, and when I graduated, E3 was the new building. So things have changed just a little bit on campus in 25 years. But I maintain a close link to Southern Ontario. In fact, get together with a bunch of buddies every year from my graduating class. So I've kept close ties with Canada, even though I've been off in California for about 20 years up until last summer. So I'm going to talk a little bit about my career pathway, but mostly I'm going to focus on the technology and I'll try to show how an engineering background has been most relevant to advancing the technology of medical devices, and now more recently in the last 8 years, regenerative medicine therapies, which I think are one of the new frontiers of medicine that are truly going to change the way we treat many diseases. So thanks for the kind introduction. I graduated in 1990, Highway 90, in civil engineering, and at the time, biomedical engineering was a department at the hospital that fixes oscilloscopes. It wasn't a multidisciplinary function that was seeking to apply engineering principles to medical challenges. It has evolved an awful lot over the last 2.5 decades. When I was in second year, I heard about this weird class called orthopedic bioengineering, which was put together by Wayne Broadland, who's my master's supervisor, and John Medley and some others, to try to come up with this cross-disciplinary approach to focus on intractable problems. And it was fascinating to me because when I met with my high school guidance counselor, they said, well, what do you want to do? And I said, well, I don't know. I really kind of want to be a doctor, but I also really like engineering, and I don't like the hours that doctors have to put in, so I think I'll apply to engineering. I mean, it was that simple for a high school kid. And when I realized that I could apply engineering principles to medical problems, well, that was just fascinating. I mean, it was really terrific, and I wondered, can I make a career out of this? I remember sitting down with Wayne one time after I'd committed to doing a master's with him, and I said, Wayne, what am I going to do with this? Where am I going to work with this orthopedic bioengineering degree? And he said, Mike, honestly, I can't tell you, I don't know. But he said, if you're good at what you do, there will always be a place for you. And that has stuck with me throughout my career. Some of the best advice I ever got. So initially, the focus in Wayne's lab was on the mechanical causes of birth defects in terms of spina bifida. I'll go into a little more detail with that. And you can't criticize me until we meet later. Then you can tell me all the errors from 25 years of lapsed memory that I produced. From that, I had a chance. I was really fortunate. In Canada, we've got NSERC scholarships, and I had some money left from my master's, and I was pretty confident I'd be able to get a PhD NSERC studentship. So I had my choice of a number of different opportunities, and I decided to go to Western to a heart valve laboratory. So that basically put me on a path into cardiovascular medicine that lasted about 17 or 18 years and now has come full circle. So I started out at the Cleveland Clinic. I was dragged over there by my professor who was recruited to the Cleveland Clinic, and it was a phenomenal opportunity because the Cleveland Clinic is the #1 cardiovascular hospital in North America. So we built up a heart valve lab there and then into industry. Well, why did I decide to abandon an academic career? I actually had an opportunity after the first year in industry to go back to the Cleveland Clinic for, I'll call it a semi-academic career opportunity at the Cleveland Clinic in an applied research lab. But I was learning so much at the hospital, at industry, at that time at Baxter Cardiovascular Group, I decided to stay there. Why? Because the closer I got to the products, the closer I got to the patient, the more impact I felt that I was having. And that has not changed throughout my career. So I'll recount a little bit of my history as I go through some of the technology. Where we started was in research into spina bifida. This is the most common mechanical birth defect in North America. It's caused when the neural tube, which occurs during neurulation and early embryonic development, basically it folds up, these neural crest cells fold up in contact, and then they seal and they form a tube that becomes the primitive spinal cord and brain. When that doesn't happen properly, we have spina bifida or other more serious defects that can have significant health consequences. And that can range from what we call spina bifida occulta, you don't really know you have it, to anencephaly, which is basically the brain doesn't form, and all areas in between. This became very personal for me because I have 2 children who were born with spina bifida. So go figure, how often does that happen? Somebody does a master's in a disease that's fairly rare and then has 2 children with spina bifida. A little knowledge is a dangerous thing, but I can tell you this knowledge has paid dividends for my children. My eldest son, he doesn't have a shunt, a medical device that he would have had to have replaced 8 or 10 times in his lifetime if I hadn't been able to respectfully challenge a surgeon when he was 4 days old. So the work consisted of trying to understand what are the mechanical forces involved at the cellular level that can cause these shape changes in large tissues or groups of cells. What you see on the left is the neural plate deforming and rolling up into a tube to form the primitive spinal cord and brain. In the upper right is a little cartoon, and this comes out of my master's thesis, so, yay for graphics from 25 years ago. What you're seeing there, and I'm sorry it's so small, are all of the little structures inside of a cell that cause it to deform, contract, tensile elements. You've got to maintain volume. Well, what can happen is if the elements around the top, these apical microtubules, if they contract, then it causes keystoning in the cell. That's a single cell that goes like this, and when you have a sheet of cells all doing that, it causes them to roll up. So we're starting to understand about the basic underlying mechanics. Now, this is the only slide that contains any graphs or, importantly, equations that you're going to see from me, because I've done everything I can to get away from the theory and onto the applied practical over the last 25 years. But my master's work involved using tensor product B-splines to model complex geometries of the surface of the embryo so that we could form a basis understanding the geometry numerically. And a colleague of mine, Dave Clausey, who you may know is a professor in systems design engineering, he did the mechanical piece, the mechanical modeling of those cells based on this geometry to understand the forces involved and what might be going wrong at the cellular level that could cause defects like spina bifida. So you can see how you go from 1D to 2D here. That's a rendered surface circa 1991, 1992. Where you had to write all the software yourself. Anybody remember OpenGL? That was a long time ago. But what we could do is characterize numerically these surfaces, which could be used as input. And what was the output of all of this? It was a degree, importantly, which is a stepping stone. And of course, Wayne has spent the greater part of the last 2 decades advancing this science and making very meaningful contributions. In fact, we made a seminal discovery together that has had a big impact on the field. I then moved into cardiovascular medicine, and this is just a brief primer. So what we're looking at is a cross-section through the heart. You've got the right side of the heart on your left side and the left on your right. And this super high-tech animation is showing you how venous return comes into the right side of the heart. And what I want to point out to you is the heart valves, because these heart valves are critical for proper valve function, heart function. And if they start to become diseased, then you can have congestion, you have stenosis, you're going to have a less efficient pump. They look like pretty simple structures. They're basically one-way check valves. They're made of biological proteins like collagen and elastin, glycosaminoglycans, and other materials, but they are incredible biomaterials. Nothing that man has been able to design in the last 50 years can come close to the durability, hemocompatibility, strength, and fatigue properties of this biological material, due in no small part to its ability to self-heal, but it's just a remarkable structure. So I got fascinated with this, and in particular I was looking at the aortic valve, which is the one you see in the middle there. That's the valve that separates the left ventricle, which does most of the pumping for the heart, from the whole systemic circulations except for the lungs. So the left side of the heart, it becomes diseased more frequently, as do the left side valves, because it's pushing pressures about 7 times higher than the right side. The same volume, right? volume in, volume out has to be the same on the left and right side of the heart, but the pressures are 7 times higher on the left. So you're obviously stressing those tissues more. They become diseased much more frequently. So this is when, during diastole, when the heart is at rest and the ventricles are filling. So the valve, the mitral valve, which goes down into the left ventricle, is open, allowing blood to flow into the left ventricle. It's then going to contract during systole, and that aortic valve is going to pop open, allowing blood to flow out to the brain, the trunk, the limbs. I became very interested in the mechanics of the aortic valve. And when you cut it open longitudinally and spread it out, it has these 3 cusps. Those 2 little holes that you see are the coronary sinuses. They importantly feed blood to the heart. And these little floppy flaps of tissue are what keep us alive. Most of us will live for 70 or 80 or more years, and our valves will never become significantly diseased. They'll just function. But these are tissues that are 1 millimeter to 2 millimeters thick that will undergo billions of cycles, that will see stresses in the order of 1 to 2 MPa, which doesn't sound like a lot until you start thinking about a 1 hertz cycle, day and night, for your entire life. So taking a look at the aortic valve, if we cross-section it, you can see there's this interesting structure of circumferential fibers. Again, these are biological proteins, collagen and elastin, glycosaminoglycan materials, interstitial cells. It's really quite a fascinating set of, sorry, I'm just making sure my timer doesn't go back to sleep so I can stay on track here. What we have is a phenomenal, interesting composite structure. You have a layer on top called the fibrosa, which is this wavy one, which is highly collagenous. And then the layer underneath, which is ventricularis, and it's mostly elastin, which is a highly, as it sounds, elastic material. And together, this allows the structure to carry significant tensile loads in the circumferential direction. It stiffens up fairly quickly. But in the radial direction, it can stretch and open up and balloon out in order to seal with its companion cusps. So a phenomenal, interesting structure. And when we look at this under circularly polarized light, we can see the collagen. And this sort of rigid, these striations show you the periodicity of the collagen fibers. So we can look at and characterize the amount and location and orientation of the collagen and the elastin, and look at the intra and interconnectivity. So my PhD thesis was involved with formulating the structure-function relationships between this complex geometry of biological proteins and mechanical forces produced by cyclic blood pressure loading. So with the freshly minted PhD, I started to work for the Cleveland Clinic at Baxter Healthcare in Southern California. And Baxter at the time, you've heard of Baxter, I'm sure. They're a large multinational medical products company. They had acquired this heart valve business, and they really didn't know what to do with it. They were used to selling bags of saline and different hospital supplies, things that you could make good money on, but you didn't have a 5 or 10 or 15-year liability after you sold it. And that was at the time of the breast implant litigation, where Dow Corning just about went out of business. So they were very scared. They didn't know what to do. So they ended up spinning the company off into Edwards Life Sciences, which was good or bad depending on how you look at it, because Edwards has become a phenomenal company worth $2 or $3 billion. But the focus here was on aortic valve disease. And there are a number of underlying etiologies that cause aortic valve disease. There are congenital defects like bicuspid valve, which is the one just to the right of the healthy valve. And then there's stenosis. Your body often responds to damage by calcifying. It's when your body sees, for example, damaged collagen fibers, it will try to repair them. But if it can't repair them, then it may form the nidus of mineralization. And what can happen over time is that they become rocky, full of calcium mineral, and they stenose. And they can be closed, they can be open. There are a number of other underlying etiologies, but the end result is you have to take the valve out or otherwise replace it. Now, more recently, you may have heard of percutaneous aortic valve replacement. This is where we go in with a catheter. That's typically mostly in sicker patients. The gold standard is still full sternotomy or a partial thoracotomy, accessing the valve directly and surgically excising it and replacing it. And I got a number of examples here you're welcome to look at after the talk. So what we do is we transect the aorta, go inside and cut out the diseased valve, and then sew in a replacement. And you can see all of these sutures, these are interrupted sutures that run all the way around the annulus of the valve, and then they come up through the valve sewing ring, the valve is dropped down in and sutured in place, and then the aorta is closed. So here are some examples of artificial heart valves. And this is an area where biomedical engineering in its infancy made some of the greatest strides in the entire industry. Initially, you had a dire unmet need. Patients were dying from heart valve disease. You had physicians, typically surgeons, who were desperate for solutions, trying to come up with surgical responses to this disease. But they didn't have a functional valve. So they tried all kinds of different things. They tried taking the patient's pericardium, the sac that surrounds the heart. They tried taking porcine valves and making valves out of other animal tissues, and these things didn't last. They might last a few weeks, a few months, but then they would deteriorate, they'd shrink up, they'd be calcified, and the patient would get sick and die. So they got together with some engineers, and the engineers said, oh, valves, I know how to make valves. I'll get you a check valve. And we started with the ball and cage technology. And the first real collaboration was with Albert Starr and Lowell Edwards, who was an engineer who had retired and decided he wanted to solve some medical problems. And it was literally machine a new part in the lab, sterilize it, put it in the patient the next day. Find out that it clotted up like crazy and the patient died. Try a new material, try a new surface finish, do it again. And over the course of a couple, 3 years, they came up with the first successful prosthetic heart valve. That's the one in the bottom middle, that's the Starr-Edwards Celastic ball and cage valve. And over the years, we developed other mechanical valves. We like mechanical valves. Why? Because we understand them. We understand the mechanical properties of these materials. We can characterize them. As engineers, it's really nice, right? We can make a little piece of material and it generally responds fairly linearly. We can make the tokens any way we want to look at wear properties. It's fairly easy. That doesn't necessarily mean that it interfaces properly with the biology. And that's one of the things that we learned when mechanical valves started to be put into patients is they're not very hemocompatible. Blood is a remarkably complex fluid. It's biphasic. It responds to shear stresses. It responds to surface chemistry. And we found fairly early on that people would get thromboembolisms, the valves would clot up if we didn't anticoagulate them. That has a direct impact on quality of life because now every time you take an impact, you bruise. If you get cut, you don't stop bleeding. So people who receive a mechanical valve generally can live with the same valve for their entire life, but they have to step down their activity level and their quality of life suffers because they're on blood thinners. And even 25 years after I started in this business, we don't have great blood thinners. It still means a significant impact on quality of life. They also can make noise, which depending on whether you find that soothing or annoying can be a good or a bad thing. So there are also bioprosthetic heart valves. Now these are valves derived from biological tissues, but if we were to take a heart valve, like the one at the top, a porcine valve, out of a pig, cut it out, and sew it into a patient, what do you think would happen? Any idea? Rejection. Xenograft rejection. Fulminant response. That's what happened in the first valves before engineers and biochemists got involved. Because those tissues were sterilized with, let's say, mercurial solutions, but they were not stabilized. So Alain Carpentier, a famous cardiac surgeon out of Paris, came up with initially formalin fixation, which was not so good, and then glutaraldehyde-based fixation. And that ended up being the ticket. And we've got about 3 decades now of optimizing glutaraldehyde fixation technology in order to improve it. That's basically tanning. It's making leather but in a very controlled way with animal tissue. So the pros are that it's a highly hemocompatible material, so you don't need to be on blood thinners. They're quite durable, although not as durable as a mechanical valve. So they'll last 10 years if you're young, 20 years if you're older. So if you're an elderly person and you need a heart valve, you get a bioprosthetic. You don't have to worry about blood thinners or bruising or bleeding. When you're younger, it's a bit of a toss-up. You have to decide, how active do I want to be? Am I willing to sacrifice some quality of life to live longer? Or would I rather get a bioprosthetic valve, which may get me 15 years, but I have to go under the knife again, which is obviously not a pleasant experience. Now, with the advent of percutaneous catheter-delivered valves, we're starting to change that. So you may be able to get 15 or 18 years out of a surgical valve, which is the gold standard, and then have another valve delivered via catheter and give you another 10 years. But the really cool thing about the valve in the bottom middle there, that's a bovine pericardial valve. And the cool thing about it is that it's made out of an engineered biomaterial. Engineered biomaterial being glutaraldehyde fixed pericardium. And one thing that I learned early on was that when you take a biological tissue and you cross-link it with a chemical, it fundamentally changes its mechanical properties. It becomes a different material. It's now engineered, and we can engineer it. We can tweak the stress-strain relationship. We can make it isotropic, anisotropic by controlling the fixation conditions. And that provides us with a whole host of opportunities for both academic research papers and improving the state-of-the-art in bioprosthetic valve technology. So on the left, you can see a typical uniaxial stress-strain relationship for bovine pericardium. And what jumps out at you? Highly nonlinear, highly variable extension. Those two things make it fairly tricky to handle this type of material. So I was fortunate to be able to work with some seminal scientists in the area in developing the constitutive relations for these materials so that we could better understand them and better model them. As you can see, what now appears to be a primitive model in the lower right corner. That's a finite element simulation of valve opening and closing, looking at different mechanical properties of this bovine pericardium. So it was pretty cool. I got to sit on a couple of PhD committees over the years and produce a number of academic publications because we needed to push that science ahead so that we could make better heart valves. We needed the science, and so fortunately, I was working at a company that saw the value in that and allowed me to set up these collaborations so we could move it forward. So these two valves on the left are a couple of the fruits of my labors over about 10 years. In the upper left, what you see is the Magna Perimount ease valve. And this is the biggest selling heart valve in the world. It's been placed into hundreds of thousands of people's chests. I take a great deal of pride, but I get zero royalties because I was an employee. I wish I worked at the University of Waterloo. I love your IP policy. But I get a lot of satisfaction knowing that this valve, and there were 3 of us on the patent for it, and I was the technical lead who took this throughout preclinical development into clinical trials in Europe and the US and ultimately commercialization. This valve lasts over 20 years in patients. It's moved the needle ahead. It provides improved hemodynamics, meaning better blood flow characteristics in especially patients with very tiny aortas, little old ladies who would be suffering after a valve replacement because they couldn't flow enough blood to get back to a normal level of activity. During a second stint at Edwards, I worked on the valve in the lower left called the Intuity, and this is a hybrid valve that's combining elements of a stent at the inflow aspect for rapid deployment of the valve into the aorta. These patients have to go on heart-lung bypass. And so the sequelae, the clinical sequelae or complications associated with going on bypass are related to how long you have to be on the heart-lung pump. So if you can reduce that time, you can have a meaningful impact on quality of life for these patients. Plus you can reduce costs because you don't need the OR for, anybody know how much an OR costs? Well, at least in the US, $3,000 to $4,000 an hour. So if you can cut half an hour off of a procedure, that's a real meaningful reduction in costs, so there are economies there, and you can get more patients into an OR in a day. So this on the left is as a repair. So we don't always have to cut out and replace a heart valve, especially in the case of the mitral valve, the valve that separates the left atrium from the left ventricle. It's often undergoes different disease processes than the aortic valve, and it is often repairable. So the surgeon will go in and reconstruct this valve, and you can see this in the lower left corner, where he's resected certain elements of the anterior leaflet and then sutured it back together, and then reinforced it with an annuloplasty ring, a ring around the annulus. The plasty is the trimming and reformation of the valve. And this has remarkable benefit, and this can last the lifetime of the patient. An effectively performed surgery by an expert can actually last as long and return the patient to a normal target lifespan. The Intuity valve, which is shown in the upper right there with its cloth cover on it now, also introduced to me delivery systems, technologies used to deliver a device into a distal portion of the body. And so in order to actuate that stent, we needed a delivery system that could go in and then expand it. And that's what you see in the lower right corner. So I'm going to take a break for 1 minute while you watch this video of how this heart valve is actually deployed in the aorta. So what you can see are 3 guiding sutures which are placed down in the annulus. The surgeon has still had to go in and cut the valve out. They drop it down in. They use these sleeves to hold the valve in place. Then they advance the deployment balloon. They set the pressure, increase the pressure, and hold it. And this will expand the stent below the annulus, so lock it into place. They then cut off the deployment system and remove it. And they use a knot pusher to push the valve down. And that's how the valve looks when it's operating during normal systole and diastole. So when I was at Edwards, I was really enjoying the work, but Edwards was a medium-sized company that had spun out of a multinational, and it thought it was a really big company. And it was a political nightmare. And I was just climbing up the ladder. I was in middle management. I had all the responsibility and accountability. I didn't have the authority. I was getting bludgeoned publicly in meetings. I mean, figuratively, of course. It was a really tough time in my career. I learned probably the most about politics and working in large organizations there, but it was not a pleasant experience at times. Knots in your stomach, getting flailed by corporate VPs. And so I was starting to get a little disillusioned with big company, big corporation, big corporate management. And so I jumped into a startup company called Orcus Medical in an exciting and area of huge unmet need called congestive heart failure. So I'll just talk a little bit about heart failure. It's a disease family, really, which is caused by at least a dozen different underlying etiologies. Heart valve disease is one potential cause, but hypertension, which is a peripheral vascular disease, can also cause it. There are a lot of contributors like diabetes, obesity, and pulmonary diseases. And there are things like heart attacks. So a lot of different things can cause it, but they all result in one thing eventually. If the disease progresses to a certain point, it becomes a downward spiral. You cannot climb back up. It's a one-way degenerative condition. And this can take quite a long time. This may take a decade or more, depending on the nature of the underlying disease and its progression. But the fact is, it goes on chronically. And it's a condition that you don't get out of. Maybe we will soon. I'm going to come back to this at the end, full circle, where I talk about regenerative medicine for congestive heart failure. So when I worked at Orcus, we were working on this system, which is a blood pump system. You can see it here. Here's a battery, a controller, vascular grafts, the blood pump itself, which together are intended to increase flow in the descending aorta. I'll show you a graphic for that in just a minute. And this is a chronic implantable device that patients would carry around to improve their cardiovascular performance, and they could move back to a normal quality of life. This is chronically applied. But what is also characteristic of heart failure is many patients go through what they call acute decompensation, where the medications that they're taking are just not adequate, and for reasons we don't fully understand, they crash. They go into acute decompensation, and if not treated or emergently, these patients will die within a few days. So we also developed a cardiology-based system, catheters, and that's this guy right here, which is called the Cancion system. And this would go up through the femoral artery and in the axillary to, whoops, yes, I got it right, increase flow during the descending aorta. And I'll show you an animated cartoon here in just a second. So this was a revolutionary thinking. We want to avoid getting to rescue therapy, which is where patients get total artificial hearts or heart transplants. We want to intervene early so that we can maintain a quality of life before patients are so far gone. So this is what the Orcus system was doing. You can see this kind of sluggish movement with the bloods moving forward and then back in the aorta. But with the Cancion pump in there, you see continuous flow, and that tickles the endothelium of the aorta. It releases nitric oxide, vasodilates the periphery, and increases venous return and cardiac output, importantly without increasing return pressures. So wedge pressure would drop, but cardiac output would increase. Nobody had ever shown this before. The basic mechanism of action is that we unload the ventricles through this flow, by decreasing blood pressure. It shifts the Starling curve towards normal. We improve renal function because we get better flow in the kidneys. Patients start to lose liquid through diuresis. And then this reduction in systemic vascular resistance through nitric oxide and other mediators results in improved cardiac output with lower pressure. And we saw we had 176 patients in our US clinical trial, and the vast majority of those patients, 90+%, were recompensated on the device for a period ranging between 24 and 48 hours. So this is what the system looks like. You've got an inflow cannula that draws blood out of the femoral artery, pushes it up through this pigtail cannula up to the top of the aorta and drops it back down. So you're basically creating a racetrack and increasing flow in the descending aorta. And most of you who know anything about fluid mechanics would say, how the heck does that benefit the heart? Well, there are a couple of mechanisms. One is a bit of a Venturi mechanism at the top, and the other element is this vasodilation, which causes a reduction in systemic vascular resistance. So there are all of the components, and you can see that's the flow circuit. Sorry, the dots are pretty tiny. Hopefully you can see this, the flow going through. So that's a cardiology-based system. These are what the components look like. So each one of these is considered a medical device. Every component of a medical device system is a medical device. Every single component has to go through full design controls. That's a US quality system regulation, the equivalent in Canada, ISO 1345 in Europe. It's a whole set of regulations. If you think that you have to deal with regulations with grant writing, you ain't seen nothing until you get into a regulated medical product. So I have, by necessity, become a design control expert over the last few decades. This is the Accelera system, so it's fundamentally the same therapy, but applied on a chronic basis, where this little implantable pump, and I've got it right here. So this is the size of the pump. This is a typical pacemaker. And there's a defibrillator pacemaker can, to give you an idea of the size. So very tiny, and yet adequate to keep these patients out of acute decompensation. We got to the clinic in 2 patients in Germany, and then 2008 happened, and the company ran out of money. And we couldn't, in good faith, go into more patients, not knowing that we were going to be around to provide technical support. And over about an hour and a couple of pitchers of beer, I could explain the whole sad story. But the end result was that technology got bought up by an 800-pound gorilla and put on the shelf because they didn't know if it was complementary or competitive. And it's not benefiting people. And that, to be honest, is the saddest part of my career, because we made phenomenal technological leaps and bounds over the course of 6 years. We got into the clinic, we showed that it worked, and then it went away. So sometimes now when I hear conspiracy theories about the perfect carburetor fuel injection system, I think, well, maybe there's something to that. So I already showed you this. I think we're doing okay on time. I'm on track. I'm not going to keep you away from lunch. And I'm glad that, some people think it's only academic institutions where food is necessary to draw a crowd. It's not true. It works everywhere. So this brings me to a point where I moved into regenerative medicine. And you might think, well, what the heck? How do you go from cardiac devices to a cell therapy? Well, I got a call from a recruiter off of LinkedIn. And this gentleman was recruiting for a VP of R&D, of device R&D at this regenerative medicine company. And I thought, well, that's fascinating. The team at ViaCyte was incredible. They were working on a treatment for diabetes, type 1 diabetes. Now, type 1 diabetes occurs, it's typically caused by an autoimmune condition where the body's own immune system, through triggers we don't fully understand, attacks its own beta cells. Beta cells are the cells in the islets of Langerhans that release insulin in response to increasing blood sugar. And when those cells die, your blood sugar shoots up, and it's a death sentence, except for insulin. Insulin was a Canadian discovery in London, Ontario, for Sir Frederick Banting. Everybody knows his name, right? Banting and Best did the work at U of T. They formed the basic science that identified insulin as this therapeutic compound. Now, we've gone a long way since then, but we're still treating the symptom of a disease. We are still keeping people from not dying. We're not curing them. It's not a functional cure. We're keeping them from dying every day. And if they get the dose wrong, they get sick or they die. It's a very serious condition. That being said, we have tried to push the needle through medical device development. We've got insulin pumps, which can titrate the dose on an ongoing basis, so you don't have this bolus of insulin with a drop in blood sugar and then a trail down as you metabolize it. So we have some improvements. We've done cadaver transplants where we take islets out of a dead person and we implant them. Now we have to give that patient immunosuppressive drugs so that those cells aren't rejected, but they can return people to an almost normal quality of life. That is kind of a remarkable proof of concept for the therapy that ViaCyte has developed. What you see on the left are human islets that have been transplanted into a patient and grown, and they function, and they can normalize blood sugar. And on the right are these engineered cells, these differentiated cells that are actually turning into beta and alpha and gamma cells in vivo after a maturation period. And the way that this is done is through what we call directed differentiation. And I'll point you to the ES, that's the embryonic stem cell, which about 15 or 17 years ago was discovered. And now since then, we've got induced pluripotent stem cells, which made a big splash. It was kind of a joint invention by some researchers, Tanaka in Japan and at the University of Wisconsin. That's where we can take an adult cell and push it back up that differentiation tree to make it pluripotent. Pluripotent meaning it has the potential to become any cell type in the body. And where the science comes in is this is a pretty remarkable biology, is understanding how you can coax these groups of cells through specific intermediaries along the differentiation pathway until, in this example, they become pancreas cells, pancreatic progenitor cells. And then once they go into the body, they are already committed to a pancreatic fate. They continue down that track, and they become a therapeutic cell type. But again, what happens when we put the porcine tissue into a patient? It's rejected. What if we put human cells into a patient? What happens? If it's HLA matched perfectly, the cells would likely survive. But you'd have to have 200 different source types to match every person in North America. So it would be rejected. So what do? We develop an immunoprotective medical device, an implantable high-tech tea bag, which is differentially permeable. It allows oxygen, glucose from the blood sugar, and other important compounds into the cell population. They can then metabolize and then generate insulin, which then diffuses out. And another very important element is that this thing needs to vascularize. So what we start is with what you see, PEC-01, that's the pancreatic endoderm cells, the pancreatic progenitors. And over the course of 2 to 3 months in vivo, they become effectively islets of Langerhans with alpha, beta, gamma, delta cells, all developed in neighborhoods, all interacting with each other biochemically and producing insulin. The device has a number of requirements. It's a safety issue. It retains the cells so that they can't spread out through the body and go rogue. Now, since they're immunogenic, they're probably going to be destroyed, but it's another layer of safety in an innovative therapy. It means that the therapy is removable. If something goes wrong, it has to be biostable. And it has to keep host cells out so that they can't get in and destroy, attack your implanted cells. I mentioned vascularization. What you're seeing here is a fine mesh on the surface of the device, and underneath that mesh is a thin membrane. This is the membrane, the magical part that has just the right porosity to allow nutrients through but not cells. After just 4 weeks, you can see these blood vessels growing along the top. Why? Because we're using the built-in biology in the cells. How do you think that blood vessels grow in your tissues from the time you're conceived? It's because all the cells in your body can identify their environment and respond to it. If it becomes hypoxic, they generate growth and angiogenic factors, basically sending a signal out to the developing vasculature, hey, I need oxygen, you need to grow in here. Adult cells remember that. They send out similar cues. These are embryonic cells, so they're sending strong growth factor and angiogenic factors that are drawing in this host vasculature. And look at this beautiful integration of host blood vessels on the outside, critical to effective mass transport between the patient's blood getting that insulin back out into the vascular system. And this is what it looks like after about 4 months. So what you've effectively got is a pancake islet of Langerhans separated by a membrane, host blood vessels throughout, and a beautiful bionic pancreas. This went into patients starting about 3.5 years ago, and the clinical trials are ongoing. So we're seeing some very exciting results where they've seen after a year. They're seeing mature beta cells growing in these devices, secreting insulin, and it's just a matter of time. Maybe there's a second iteration that's already in the clinic. We're still waiting for a readout. That one could be the silver bullet, but it's just a matter of time before we lick this. And we'll have a treatment which is effectively a functional cure for type 1 diabetes. So that takes me to where I am now, which is a company called BlueRock Therapeutics. The company was founded in December 2016 with, I believe, the second largest A-round investment in biotech history by Bayer AG, big German pharmaceutical company, and Versant Ventures, which is a professional venture capital company. And it was $225 million, which sounds like a lot of money and is a lot of money, but it's not enough money. It's not enough money to take products through to clinical success. That being said, it gives us the bandwidth and the runway to make meaningful steps forward in a limited number of areas. And what we've decided to do is focus in a couple of areas, and it includes congestive heart failure and Parkinson's disease as our first 2 clinical targets. So we're located in 3 places, including Cambridge, Massachusetts, which should be of no surprise to anybody who knows the biotech industry, Manhattan, and Toronto. Surprised? Shouldn't be if you know the environments, but to most people they'd go, why are you, Manhattan's too damn expensive, why would you go there for stem cell work? And same with Toronto. Well, guess what? Stem cells were discovered in Toronto in the early 60s. So there's a long history of stem cell biology work in Canada. And there's an enormous untapped resource in the Toronto area without a lot of industrial collaboration available. So there is an opportunity for a company like BlueRock to come in and make some real strides. Our basic strategy is to co-locate the industrial product development engine right beside the discovery researchers. So in Toronto, that includes Keller and Mike Laflamme. And it basically allows us to work very closely, even on a day-to-day basis, with the people who develop the science. So we've not just taken a cross-functional team at the product development level, but at the discovery level as well. And this allows us to identify promising early research results and say, That's really interesting. We want to double down on that. We want to give you money with just an SRA, a sponsored research agreement that we can work out in a couple of months to go ahead and move that science forward to a go-no-go decision. Maybe a no-go decision, but we moved it faster than would happen with the normal cadence of academic research where you've got to go through granting cycles. I know you're all going, how do we get into that? Yeah. Well, the reason we went to New York is because Lauren Studer is one of the world experts at differentiation from pluripotent stem cells, including the whole central nervous system. And he, in fact, generated dopaminergic neurons that produce dopamine in response to biological cues for treatment of Parkinson's disease. And his, actually, turns out his wife is a top neurosurgeon who's now the chief of neurosurgery at Memorial Sloan Kettering. She's our physician who will be implanting the first patients with this therapy at the end of this year. So basically, Parkinson's is caused by a loss of dopaminergic neurons. These are located down in the substantia nigra, down in the deep brain. And when they're lost, you start to develop Parkinsonian symptoms. This can be neural, it can be cognitive. This therapy is intended to treat the motor deficits that are caused as a result of loss of these cells. And there's an important clinical predicate here where fetal brain tissue transplants were performed about 20 years ago, and they demonstrated long-term efficacy, 18, 20-year efficacy, even though their starting material was kind of sketchy in terms of quality and how it had been prepared, and the stereotactic delivery technology was kind of sketchy too. So we've been able to improve every category in the product, so we have very high confidence that these patients will see benefit even in the first-in-human trial. So fingers crossed, because this will be an extremely important win for the entire field of regenerative medicine. So we're headed for the clinic this year. We've already manufactured our GMP, good manufacturing practice compliant clinical material. Our preclinical models show remarkable efficacy, even using human neurons. So we use immune knockout animals, so they don't have an immune system. And then we can selectively knock out their dopaminergic neuron cells, and then we replace them with the human cells, and we can return their function. And this has only occurred through developing mastery over the cell biology associated with causing those pluripotent cells to become these dopaminergic neurons. They continue to mature somewhat in vivo, and they develop these interconnections with the host tissue. So these neurons interface with host neurons and they start communicating biochemically, and then that's when you really see the efficacy start to ramp up. And we see the lower curve, I'm sorry you can't really make it out, but you'll have to trust me, suspend your disbelief, it's a good curve. It's showing a pretty remarkable improvement in function which was lost due to the chemically induced lesion. Closer to home here, the focus is on cardiovascular disease. And Gord Keller, again, if I had to list the 3 top developmental biologists focused on differentiation biology in this space, I would choose Gord Keller and Lauren Studer in the top 3. These guys are world-renowned. And Gordon is an expert in mesoderm, all of the kind of cells of the gut and the trunk here. So he's basically figured out how to make ventricular cardiomyocytes. These are the contractile cells in the ventricle which cause contraction. In fact, he's even working on left versus right ventricular differentiation. So these cells actually contract all by themselves in the dish. And if you look really closely, can you see this? This is a monolayer culture of ventricular cardiomyocytes, and they're beating. What you can see in the middle there is those cells, once we pull out, you can see actin and myosin filaments. The different colors are human and rat cells integrated in the myocardium, functioning together. And we've got some really cool videos with these fluorescing cells. When they contract, they brighten, and you can see the host ECG lined up with this pulsing graft muscle tissue. It's really remarkable. So this technology is a little behind because it's a longer path to the clinic, and it's a very complicated because we've got to develop all the delivery systems to deliver the cells to the ventricle. We're talking microliters of cells delivered over really long tubes. So we're really advancing the state of the art in terms of medical device technology. And with that, I will call it quits and leave 10 minutes for any questions. And yeah.
So the cells that you're putting in BlueRock technology, these embryonic cells isolated from the patient?
They're not. There are two different approaches called autologous, which you're referring to, where we take a sample of cells from the patient and then treat them somehow and deliver them back to the patient. These are what we call allogeneic. The reason for that is autologous therapy is very complicated and expensive. You take a sample of tissue from a patient, it has to have a very strict chain of custody. It's very, very expensive. And then you have to have robust manufacturing processes that will work with highly variable incoming raw material. With an allogeneic approach, we can very tightly control the incoming cell type. We can fully characterize it, the manufacturing processes, and put out a very high quality product. But what we're doing is performing what's called HLA editing. We're editing those cells as pluripotent cells to eliminate the immune response. And it's a little more tricky than that, but basically we can knock out certain genes, knock in others, and then the cells will look like self, so they won't be rejected.
So you're going to modify the cell and put it back into an individual. So you have this separate group of stem cells, you've modified them, and then you put those into it.
Yeah, the stem cells themselves don't go into patients. And here's a take home. Don't believe everything you read about the magic of stem cell therapy. Stem cells are remarkable, and we have them in our bodies, different types of stem cells, not fully pluripotent, but different types, like hematopoietic blood stem cells, for example, in the bone marrow that are constantly generating red cells and white cells. But stem cell therapy is a misnomer. You don't just put pluripotent cells into a patient. I call that the reverse field of dreams. That's like squirting something in and figuring those cells are just going to know where to go, and magically they're going to go there and they're going to do the right thing. That's ridiculous. It's specious, right? They're going to respond to their environment. They're going to make teratomas. What we do is we use those as source material, perform these directed differentiation operations on them until they become specific targeted cell types with a known mechanism of action delivered to the right anatomic site. Yeah.
Don't you still need to take a sample of the person to find out what you're going to need to do to the host cell, your stem line or your line, to get them to not be rejected by that person? Do you have most of the same problems?
Well, immunology is a very complex field, but we do understand enough. There's what we call HLA typing. So there are certain classes of antigens that are present on the surface of cells. And as I mentioned earlier, there are about 200 different kind of unique classes. If we were to have 200 different allogeneic cell banks, we could potentially take a sample from the patient and HLA match them. That's what they're doing in Japan, but they only have about 17 HLAs in Japan. In North America, we have to deal with the whole world. So what we have done instead is we engineer the cell so we don't have to do that. And what we do is basically knock out the surface markers that identify the cell as non-self. And then we have to actually put in some, otherwise a different part of the immune system, the innate immune system, will identify this as an abnormal cell because it doesn't have those markers and will kill it. So we have to basically stealth the cells so that they fly under the radar of the immune system.
Doesn't that make them look a lot like cancer cells?
That's why we have to knock back in those surface markers. So they're specific antigens that are identified by the innate immune system cells, like giant cells, for example, so that they don't get rejected. Something over here?
Yeah, I was just wondering if, from your perspective, you work for a few different companies and different medical devices. How would you compare, in general, your work with the different medical devices as compared to a non-medical product that's similarly complex and similarly technologically advanced?
Well, not having had the benefit of working in some of those other fields, it's a little difficult to say. But what I can tell you is, since I started with applying an anticoagulation coating to an annuloplasty ring at the beginning of my career to this regenerative medicine approach, I have progressively gotten into more complex and more multidisciplinary programs. And so the complexity and challenge of the science and the engineering has increased, and the regulatory environment hasn't gotten any easier. They constantly layer on new requirements. So in the medical device field, because there is such a desire to protect patients from evil, greedy companies, you have to jump through a lot of hoops in order to get the products through. And so a while ago, I think after Orcus Medical failed, I said, you know what, screw that. I am not going to go with the most challenging brass ring, cure every patient on the planet type therapy. It's too hard. It's too disappointing when you get close and it fails for reasons that weren't the science. But each time I change jobs, I jump into a more complicated, more challenging, more difficult. So that tells you what kind of stimulating and incredibly rewarding experience it is. I would say that while other fields are increasingly cross-disciplinary, the field of biomedical engineering is by far, in my experience, the most cross-disciplinary. Where else do you learn about stem cell biology, biomaterial stability, interactions between your cells and the materials, vascularization from the host, all of the variability of biological systems, which would just be frustrating to all of you. They're frustrating to me. Take everything you know and multiply it by variability of 10. It's plus or minus 20% patient to patient. Fascinating, challenging, but I think the variability and the cross-disciplinary nature are two things that would distinguish biomedical and medical device technology development from many other fields.
So the regulatory system is this massive wall that any company has to crawl over, and it stops a lot of people from even trying. What do you see some changes coming to that? I understand what it's supposed to do. I understand why it's there and why it may be necessary. But do you see some hope for some changes coming down the pipe or something?
There have always been ways to game the system. There's a humanitarian device exemption, which is a way if you've got a fairly limited patient population at high risk where you can leapfrog around a bunch of the early stage requirements. In Canada, there's a special access program for medical devices where a physician with a patient in an intractable condition where there is no other therapy can prescribe a custom device. And so you can basically get that product. And that's how Edwards Life Sciences developed the percutaneous valves and delivery systems. They did it out west in Vancouver, and they iterated like every 3 or 4 months, unheard of cycling of product iterations in order to move the needle and be successful with that. And they did that through the Canadian Special Access Program. Most people think Canada is impenetrable. It's really not if you use the system that way. Now that, you might argue, is stretching the intent of the special access, but at the same time there are ways. But you're right, conventionally it's very difficult. There are very high bars, and the regulators play basically quality assurance cop. They're like, no. You prove to me you're not going to hurt anybody. You say, well, I can't prove to you. I haven't been in any patients yet, but I can show you with a high degree of confidence that the risk-benefit ratio makes sense. There are programs in place to try to accelerate development. There are fast-track designations. There's something called RMAT, which is Regenerative Medicine Advanced Therapies option, where, again, if there's a significant unmet need and you've got some exciting technology and even just a whiff of preclinical efficacy, that you can move more quickly into the clinic. What that doesn't alleviate is the high quality bar for once you get to a phase 3 clinical trial. So what you could end up doing is getting quickly into the patient, which is important, getting those clinical learnings. And if you're successful with that accelerated program, then all of a sudden your manufacturing team and your assay team have to catch up because they just got their timeline to commercial activity shortened by a year or two, which is a great problem to have, but they haven't taken the pressure off the sort of end stage commercial requirements. Yeah. We are at 12:01. We hit it on time.